So last time we compared the size of bacteria to other types of cells. Bacteria very, 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 very small. And in the lab, we could compare the bacteria to our cheek cell size, so that was really great. Another aspect of bacteria we looked at is how do they move around? So what is the structure called with which they move around? So you can type that in the chat if you like, or you can unmute. The flagella, yes, the flagella. So that's what they use, and it kind of moves in a whip-like motion so that the bacteria can move around to areas of better food and away from danger. So if moving toward or away from something, what is that called? It starts with a T, if you remember. And it's kind of like hailing a cab. That's not a cab. A taxi, yeah. Good. Taxis. Yeah, so taxis are moving toward or away from something. There was phototaxis, toward or away from light. Magnetotaxis, chemotaxis. We did talk about the teeny little plasmid circular bits of DNA that bacteria have. What's interesting about those is they give us the capability of producing things such as insulin. And we also discussed reproduction. So bacteria reproduces asexually and sexually. So mostly asexually by binary fission. And they also can exchange DNA through these pili. So the little plasmids would travel from one bacterium to another. And what's fascinating too about bacteria is that they exchange DNA not only between the same species of bacteria, but also different species. And that is also one of the reasons why bacteria are so diverse. So classification in bacteria is a little complex, but there are basically two domains in which prokaryotes exist. One domain is called Archaea. Those are your extremophiles. And the other domain is just simply called bacteria, or some people call it eubacteria, EU bacteria. And there are various groups within it. We'll look at some of the different groups. And somebody's mic is on. If you wouldn't mind just muting while the lecture's on, unless you want to ask me a question. The domain Archaea includes the nanoarchaeoids, and we aren't going to get hung up on the exact names of each group. So what I find interesting is what bacteria do in the environment. So how do we classify them? They're really, really small. To an untrained eye, they pretty much all look the same. So the differences are, we've already seen their shape, their cell wall composition, so whether they're gram-negative or gram-positive. And we can also look at their DNA. So there's a method known as polymerase chain reaction. And that is a way of sequencing genomes. Genomes are all the genes within one individual. The human genome was sequenced in 2003, around there. I think it took 10 years. Now you can sequence DNA in a matter of hours, and sometimes less if it's a small genome. Yeah, so we know that there are many species, and the exchange of DNA via the pili is known as horizontal gene transfer. So it kind of obscures the root of the tree of life of bacteria because they are exchanging DNA. So these are some of the groups: Proteobacteria, gram-negative bacteria. They're very diverse. So here's a Proteobacteria called Rhizobium. It lives inside the roots of trees, and it fixes nitrogen, which is very useful for the tree. What's another one you may have heard of? The one at the bottom here is Helicobacter pylori. That is a bacteria that lives in the gut of humans. About 50% of us have it, and now it's been discovered that it's the bacteria that can cause ulcers, given other conditions as well. Some other groups are the chlamydias, the spirochetes, gram-positive bacteria, and cyanobacteria. Chlamydia, which live inside animal cells. Here's a spirochete that you might find in an aquatic environment. Streptomyces, that's gram-positive. It's the source of many antibiotics. There are the cyanobacteria, and they're extremely common in freshwater and in saltwater as well. Archaea, one of the domains of prokaryotes, share some traits with bacteria, and they share other traits with eukaryotes. So I encourage you to look at this table. It's a table of comparison of the domains of life, and it gives you the reason why those three domains are the broadest generalization of all life on Earth. Yeah, so they seem like simple things, but at the cellular level, these things are like super important. And if you have the technology, you can see those differences as well. So, for example, I'll just point out a couple. Nuclear envelope, absent in bacteria and archaea. They're prokaryotes, they have no nucleus, and they're present in eukarya. Peptidoglycan in the cell wall is only present in bacteria, not in archaea, and not in eukarya. Yeah, so they're quite interesting. Circular chromosome is present in bacteria, present in archaea, but absent in eukarya. Although that's not entirely true, because the smaller plasmids are present in mitochondria. But mitochondria, according to the endosymbiont hypothesis, are bacteria from ages ago that have been engulfed by other cells. So mitochondria have their own plasmids, as do chloroplasts of plant cells, which were also thought to have been engulfed in the distant past. Archaea includes some really interesting species that have these amazing capabilities of survival in extreme environments. So the term halophiles refers to individuals that live in a very high saline environment. A methanogen lives in swamps and marshes and produces methane as a waste product. And the reason why bacteria can live in so many different environments is because they have different ways of collecting energy and food. So here's some terminology. Photoautotrophs, or phototrophs rather. Phototrophs, like plants, they obtain energy from light. So a plant obtains its energy from sunlight, which starts photosynthesis happening. Chemotrophs, that's you and me. We obtain energy from chemicals. So we take that glucose that's been made by the plant, we ingest it, and that's where we get our energy. It goes through the mitochondria through cellular respiration. An autotroph requires carbon dioxide as the carbon source. So plants are autotrophs. Indeed, a plant is a photoautotroph. Heterotrophs require organic nutrients to make organic compounds. So you and I are heterotrophs, so we have to eat our fruit and vegetables in order that we get our nutrients. So we can combine those to give the four major modes of nutrition. And that is photoautotrophy, chemoautotrophy. I know these are kind of cumbrous, but that's what they're called. Photoheterotrophy and chemoheterotrophy. Photoautotrophs include plants, includes multicellular algae, which is a protist, so that comes under the eukarya. Unicellular protists, also a eukaryote. But cyanobacteria is a photoautotroph. Cyanobacteria is also a kind of algae, but a bacteria as opposed to a protist. And then there's purple sulfur bacteria, also a photoautotroph. Some chemoautotrophs usually live in areas where sunlight just is not available. So there are bacteria that live in the deepest depths of the ocean, the middle of the Atlantic ridge. There is no light there. And so they gain their energy from chemicals, such as, that's a good question, such as copper, perhaps. Photoheterotrophs are somewhat rare. You don't tend to find them too much. Organisms that get energy from light, but ingest nutrients from other things. So there's a very few marine species. These tend to live in very hot places. This is a microbial mat at a hot spring. Chemoheterotrophs are those that require energy from chemicals and nutrients from absorbing from outside or ingesting from outside. So heterotrophs are you and I, so we ingest material from outside of ourselves. Saprobes also get materials from outside, but they do it by absorbing nutrients from dead organic matter after they have decomposed it with their own enzymes. Those are called saprobes. Parasites is another type of chemoheterotroph, and it absorbs nutrients from the body fluids of a host. Another reason bacteria are so prolific is that they have some interesting metabolic relationships to oxygen. In other words, sometimes they don't even need it. But some of them must have it. An obligate aerobe requires oxygen to survive. A facultative anaerobe can survive with or without oxygen. So they have the capability of respiration and fermentation in order to get energy. Carp are one of those animals. So carp live in droves at the bottom of Lost Lagoon where there is no oxygen, but they come to the surface sometimes and they can live with or without oxygen, but sometimes only for short periods of time without oxygen. So humans, of course, we cannot live without oxygen, but some of our muscle groups can for a short period of time because we also have anaerobic respiration. We can produce ATP anaerobically, but only a small amount. Obligate anaerobes are poisoned by oxygen. And it's thought that a long time ago, 3.8 billion years ago, which is when the first bacteria are discovered to have been on Earth, and some of that discovery is from Canada, from the Great Lakes. Traces of bacteria have been found, but also from microbial mats in, no, they're not microbial mats, sorry, they are calcium carbonate remnants called thrombolites, and they are in Australia, and they also date back 3.8 billion years. But during the progression of the atmosphere on Earth, it went from having very little oxygen to having a lot of oxygen. And so the first bacteria would have probably been obligate anaerobes. So when oxygen became more prevalent, they died off but the aerobes started to increase. Indeed, oxygen is poisonous to my sister's calling me in the middle of my class. What is she thinking? Okay, so one thing I'd like to talk about also is the cycling of nutrients through our ecosystems. That would not be possible without bacteria. And one very interesting area of that is called nitrogen metabolism. So prokaryotes metabolize nitrogen in a variety of ways. One is called nitrogen fixation, where nitrogen is taken from the atmosphere and converted to ammonia. And from there, it can be converted to other things, and then, and only then, can be used by plants. Plants cannot use atmospheric nitrogen. The advent of single cells was followed by an interesting phenomenon of forming colonies. So there are colonies of bacteria, and one colony is the Anabina, the cyanobacterium called Anabina. It has photosynthetic cells and nitrogen fixing cells. So the nitrogen fixing cells, well, they can take that atmospheric nitrogen, convert it for the rest of the cells in the colony. The photosynthetic cells, they produce sugar via photosynthesis, and they can share it with the nitrogen fixing cells. So that's one of the first incidences of cooperation between cells living in a colony. This is like a filamentous kind of colony of cells. So chemical recycling in an ecosystem occurs as a result of prokaryotes. They recycle chemicals between the dead and the living. It sounds like something out of Halloween. Yeep. It's not scary. It's a perfectly natural thing to occur. So this is a cycle of decay, rainfall, and tree roots taking up nutrients. And decaying vegetation quickly becomes nutrients. And it becomes nutrients because bacteria have that capability of converting atmospheric nitrogen. And they break everything down. They break down all our dead material. If it weren't for bacteria, well, the earth would be stacked up to the moon with dead bodies. Be very unpleasant. So nitrifying and nitrogen fixing bacteria. All proteins contain nitrogen. The atmosphere is 80% nitrogen, but plants can't use it directly. It's the wrong form, N2. They need it in ammonium, NH4, or ammonia. The bacteria do this conversion. Where do they live? They live in these little nodules. You can see them here, little nodules in the plant roots. Particularly the legumes, like alfalfa and soybeans. Farmers can plant these crops purposefully to enrich the soil with nitrogen. They live in soil also freely. These nitrogen-fixing enzymes, however, are poisoned by oxidants. So that's why they live symbiotically with the roots in order to remain apart from oxygen. Nitrifying bacteria can convert ammonia into nitrate. And nitrate is ultimately one of the forms plants require in order to build their own tissue and their own proteins. So chemoheterotrophic prokaryotes are decomposers. They break down corpses, they break down dead vegetation and waste products. And nitrogen-fixing prokaryotes add usable nitrogen to the environment. So we make use of bacteria for their skills in sewage treatment plants, for example. Some bacteria are symbiotic with organisms like this fish. These bioluminescent areas of the fish are actually bacteria that live in the skin of the fish and are bioluminescent themselves. So there are incidences of mutualism and commensalism abound in nature. So there are three kinds. Symbiosis is kind of a loose term, but there's really three kinds: mutualism, commensalism, and parasitism. Sorry, symbiosis is a term that's used rather loosely, but to be more specific, mutualism, both benefit from the arrangement. Commensalism, one benefits, one doesn't care. Parasitism, one benefits, usually the bacteria, and the host suffers. So prokaryotes have both harmful and beneficial impacts on humans. Some are pathogens, but most others have positive interactions with humans. So some of these are Bifidobacterium bifidum, Lactobacillus acidophilus, and E. coli. Those you will find in the body of humans, lactating females, for example. And they help to protect infants, for example, from invading bacteria. And E. coli in the digestive tract, in the large intestine, make some vitamins that we require. Other ones are Staphylococcus epidermis. Some are different species you find in the feces. Streptococcus pneumoniae, that's found in the upper respiratory tract. I think about 50% of the population has that as well, but they don't infect the lungs unless the immune system becomes somewhat deficient or a person is sick for other reasons. And we already talked about Helicobacter pylori. Yeah, so there's a lot of pathogenic prokaryotes that cause about half of all human diseases. Lyme disease is an example of that. And most of those diseases are from endospore-forming bacteria. They're mostly in the genus Bacillus, and their spores are very resistant to the environment. They may survive possibly millions of years before they revive. Anthrax is caused by a Bacillus species, and the family of bacteria that cause botulism also, and tetanus, are this type of bacteria. Some bacteria cause rickets and chlamydia. Chlamydia is a very common sexually transmitted disease. Rickettsia live in the lining of insects, and they infect mammals through mosquitoes, so there's often a carrier of the bacteria. Some bacteria that are intracellular parasites of eukaryotic cells, they can't survive outside of a cell. Some other ones, some infections like Staphylococcus aureus and Neisseria gonorrhea are two examples. Staphylococcus produces pneumonia, for example, toxic shock syndrome, strep throat, some other diseases. Typically, disease is caused by the release of exotoxins or endotoxins, and some are potential weapons of bioterrorism. But we also use bacteria for such things as bioremediation, so we can use them to clean the environment, because bacteria can sequester toxins. We can also use bacteria in such things as mining, the synthesis of vitamins, the production of antibiotics and hormones, and other products. So in this lecture on prokaryotes, we have looked at the distribution and abundance of prokaryotes, their structure and function, taxis, genetics, heredity, classification, nutrition and metabolism, and the impact on the planet and humans. And I'd like to end there for now. Thank you for watching.